Have you ever wondered how a safe and effective COVID vaccine was developed in less than a year when traditionally vaccines took about 10 to 15 years for full development? Well, welcome to the world of mRNA-based vaccines. And over the next couple minutes, I'm going to describe the general principle behind vaccination, the differences between traditional and mRNA-based vaccines, why mRNA vaccines constitute a breakthrough in immunology, and what the next steps are for this field. So without further ado, let's get started. Okay, so to understand how vaccines work in general, you need a brief understanding of how your immune system protects you from pathogens. That is, something like bacteria, viruses, fungi that want to take over your body. Adaptive immunity is a specific response against these pathogens that's sparked by an immune-stimulating molecule known as an antigen and carried out by a class of cells known as white blood cells. Under the cell-mediated response, cytotoxic T cells cause infected cells to self-destruct. Cytotoxic T cells are activated when they're presented with antigens, often through antigen-presenting cells, which engulf the pathogens and then present their antigens on their surface for white blood cells to come and recognize. Cytotoxic T cells can eliminate infectious agents themselves, or they can help other cells to do so. Under the antibody response, B cells are activated to secrete proteins called antibodies, which circulate your bloodstream and bind to infectious agents and their toxins in order to either prevent them from replicating and destroying your body, or to help other cells see them and destroy them. Another concept critical to learning how vaccines work is immunological memory. After an initial encounter with a pathogen, some immune cells will retain a memory of how to fight it. This means that the second time around, if your body encounters a pathogen again, it'll have a faster and stronger response. And this is the main principle behind how vaccines work. Okay, so now that we understand the general principles behind how our immune system functions, we can understand how traditional vaccines work. Traditional vaccines contain weakened or inactive parts of the pathogen, aka an antigen, to trigger the immune system to act as though it were really infected, to develop an immunological memory, and protect against disease if you encounter the pathogen in real life. There are a couple different types of traditional vaccines that I'll quickly outline here. First, live vaccines use a weakened form of a disease-causing microorganism in order to stimulate the immune response. Many provide lifelong protection from a pathogen, but they might not be suitable for some groups with weakened immune systems. Inactivated vaccines use dead microorganisms. They don't provide as strong protection as live vaccines, so booster shots might be necessary for durable protection. Finally, subunit, recombinant, polysaccharide, and conjugate vaccines use a piece of the pathogen, like a sugar or a casing or protein, in order to stimulate a strong immune response. Boosters still might be necessary, however. These vaccines are safe for most, even those with weakened immune systems. Okay, so now we understand how our immune system works, and we understand the principles behind how traditional vaccines work. Let's now get into what mRNA vaccines are and how they work. But to answer that question, we need to understand what mRNA is. mRNA stands for messenger ribonucleic acid, and it's a type of single-stranded RNA that's involved in the process of making proteins. In the transcription step in protein synthesis, mRNA is transcribed or made from a DNA template. The role of mRNA in the process of making proteins is to carry the protein-making instructions from the nucleus to the cytoplasm, where they encounter the protein-making machinery of the cell, the ribosomes, that read the sequence and translate it into a chain of amino acids that becomes the final protein. All right, so now with our background, we can begin to understand how mRNA vaccines actually work. In general terms, scientists will design a particular mRNA sequence that codes for the antigen corresponding to a particular pathogen. When that mRNA sequence is delivered into cells, it's translated into that protein using the cell's own protein-making machinery, those ribosomes that we talked about before. That protein is then shown on the cell surface, and it stimulates both the antigen response and the cell-mediated responses that we discussed on the first slide, which makes this a powerful vaccine system. Beyond the general idea that we just talked about, once mRNAs are engulfed inside of the cell and the escape membrane-bound vesicles, known as endosomes, they enter the cytosol. They then encounter the ribosomes, those cellular protein-making machines that we talked about earlier, and they're translated into the target antigen. A portion of the antigens produced are displayed on the cell surface, where they are presented to B cells and T cells, sparking the cell-mediated and antibody response. Another portion of the antigens produced are released outside the cell to recruit and activate other immune cells and further amplify the immune response. This dual mechanism is part of what makes mRNA vaccines so powerful. And on that note, let's discuss all of the major reasons why people consider mRNA vaccines to be a breakthrough in immunology and vaccinology. First and foremost, they have a significantly shorter development timeline than traditional vaccines. And this is what we were talking about with the COVID vaccine previously. Historically, it took about 10 to 15 years to design a vaccine, but the Pfizer vaccine was designed, manufactured, produced, and proven safe and effective in less than one. Additionally, mRNA vaccines are safe. mRNA does not integrate into the host genome and is also non-infectious. mRNA vaccines are highly effective, again, thanks to that dual mechanism that we talked about earlier, and their production is fast, scalable, and cost-efficient. mRNA vaccines also provide the ability to encode for multiple antigens, which means it's a great tool for targeting very resilient pathogens. And finally, it has a wide range of applications, from protecting against influenza to cancer. However, there are some hurdles in the field currently, most importantly, instability due to degradation by enzymes in the body and insufficient delivery. Therefore, current research is primarily focusing on improving mRNA delivery systems. For this kind of vaccine to be effective, the mRNA sequence must reach the ribosomes inside the cells. However, mRNA sequences themselves are highly negatively charged, they're large in size, and they're rapidly degraded by enzymes inside the body, as we discussed before. This means that they require some sort of delivery vehicle to protect them and carry them to cells. And one of the most popular delivery vehicles right now are known as lipid nanoparticles. And these are fat-based genetic carriers that basically envelop the mRNA, protect it from the outside environment, and deliver it to the cells. Additionally, scientists are focusing on chemical modifications to increase the stability of the mRNA sequence in order to protect it further from the outside environment. And finally, some researchers are integrating AI and CAD into designing new and improved delivery vehicles for mRNA vaccines. Thank you so much for watching. I hope this talk inspired you to check out some of these references and familiarize yourself a little further with the amazing potential of mRNA vaccines. And I'd love to know, if you were a researcher, what's the next thing that you would focus on in this field? Bye.